Now that we have gone through amino acids and into proteins, we're going to be looking at these specific proteins called enzymes. At the beginning, I want to review the different types of enzymes. You're going to see these pop up again and again and again, and it is key to understand and recognize what the function of these enzymes are. Oftentimes, when we're talking about board questions, if we understand the base function of the enzyme, that can help us get to the answer even if we are unsure about the actual specific enzyme. So here we have the way that these are broken up. The ones that are bolded, specifically the ones that are highlighted, are some of the most important ones that are most commonly going to be asked about, that you're most commonly going to see. The ones that are simply bolded are important. The ones that are not bolded or highlighted may still come up, but less often. So amylase, its job is to break down starch, break down carbohydrates. Carboxylase adds carbon onto a molecule. Decarboxylase removes carbons from a molecule. Most often, this is referring to CO2 molecules. So you will see a carboxylase, you are using a CO2, you're adding a CO2 back. Decarboxylase, you're creating a CO2, you're removing it off of it. Dehydrogenase, removing of hydrogen atoms. You have DNA polymerase, helicase, which unwinds DNA. The DNA polymerase synthesizes DNA. Hydrolase does hydrolysis. This is breaking chemical bonds using water. Isomerase, another word, we have isomer. This is rearranging the structure of the molecules into an isomer. An isomer means that it has the same chemical compound, but a different structure. Kinases, these transfer phosphate groups, typically from ATP. So they take a phosphate from ATP and they stick it onto another molecule. Lipase breaks down fats. Ligase is responsible for joining molecules together, a lot of times using ATP for that. Lyase, very similar to hydrolase, except not using water. So the hydrolase breaks down chemical bonds using water. Lyase breaks bonds by means other than hydrolysis or oxidation. Nuclease, these break down nucleic acids, DNA and RNA. Oxidase, these catalyze oxidation reduction reactions. We'll see those again in a second. Remember, we have oil rig. Oxidation is loss of electrons or hydrogens, because the hydrogens are often associated. Hydrogen is often holding a single electron, so we'll see that a lot. And reduction is gain of electrons or hydrogens. So we're either losing or gaining electrons. And oftentimes within an oxidation reduction reaction, we have one molecule is being oxidized and another molecule is being reduced, or vice versa. So one is always reduced, one is always oxidized. They go in pairs like that. This is actually another one that will be very, very common. Phosphatase removes the phosphate group. Phosphorylase adds a phosphate group to a molecule. Protease, this is breaking down proteins. A ribonuclease, breaking down RNA. Synthase synthesizes molecules. We'll call anabolic reactions. I always think of anabolic. You always hear anabolic steroids. Makes you grow big, big, big muscles. Makes things grow, brings things together. Synthase anabolic reactions are synthesizing or making stuff bigger, bringing stuff together. The opposite of anabolic is catabolic. Catabolic is breaking things down. That would be our lyases, our hydrolases, potentially our oxidases as well. Breaking things down, that's our catabolic reactions. Transferases transfers functional groups from one molecule to another. So that is all, most, I should say most, not all, of our different types of enzymes that we could potentially see on this exam. Some general information about enzymes, kind of what are they? All of our enzymes are catalysts, which means that they make things happen faster, they make things happen sooner. The way that they do that is they lower the activation energy. For every single reaction that's about to take place, it requires a certain amount of energy to be put in order for that thing to happen. Whether that be putting energy in order to form a bond, putting more energy in to be able to break a bond somehow. Everything requires some type of energy to make it happen. So enzymes, they come and they bind and they help the situation along to help have it require less energy to do so, making the reaction more likely to happen. Some important info on enzymes is that they are never consumed during the reaction. It means that they never become part of the substrate or part of the product. They're always there at the beginning and they're always there at the end. They do not get broken apart or used in any other way, or used up in any other way. They're at the beginning and the end. The substrate, we call that word, that is the molecule that the enzymes are acting on, the enzymes that are using, and the product is what is produced. We dive into this world of what is Km and Vmax and all of this good kind of stuff. Km, this is something that is typically calculated within a lab, depending on the different type of substance, depending on the type of enzyme, depending on all of these different factors. They go into a lab with a little beaker and they mess with everything, and they're able to measure the affinity an enzyme has towards the substrate. Affinity is like attraction. So it's saying how well does the enzyme attract and grab onto the substrate. So we compare affinity to magnets. So the higher the affinity, the stronger the magnet, the stronger the pull that enzyme has to want to grab onto that substrate and help it go through its reaction. It gets a little bit confusing because Km and affinity has an inverse relationship, meaning that the higher the affinity the enzyme has towards that substrate, the lower the Km value. So and vice versa, the lower the Km value, the higher the affinity. This is where people often get confused by this, where they ask questions involving this, and they often get confused and think, oh, high affinity, high Km. It is an inverse relationship. High affinity, low Km. High Km, low affinity. Okay? I want to beat that into our brains, that inverse relationship. Now we talk about Vmax. Vmax, again, this is seen, and this is something that we look at when we're putting different stuff into a little beaker, into a little vial, looking to see how quickly reactions occur. Vmax, this is the max velocity, the max speed of the enzyme reactions. So in more English type words, this is how quickly the enzymes that we have can take all of the substrates and turn them into products. How quickly does that happen? And we get this formula, Km equals 1/2 of Vmax. So these two things are related. You think about it, if we have more affinity, the greater the affinity, more likely we are to have everything happen quicker. So we'll typically have a higher Vmax. And we can actually put this in there. So Km, a really low value of Km, if you divide it by 1/2, that's the same thing as multiplying things by 2. Everything gets a little bit bigger. Divided by 1/2, Km is going to be bigger, we have a Vmax. So the larger the Km, the bigger the Km value, the lower that Vmax is going to get. That does not quite sound, I don't believe that I stated that quite properly. So I want to make sure that we all stay super clear on that. If that was a little bit confusing, don't worry about it. Don't spend too much time focusing right on that. What's really important is this next part. So there are 3 major factors that affect the reaction velocity of an enzyme: the substrate concentration, temperature, as well as pH. So if we change any of those, that is going to affect our velocity. Oftentimes, raising the temperature usually has a positive impact on our Vmax. Increasing substrate concentration often has a positive effect on the Vmax. And pH depends on the enzyme. Some enzymes work much better in low pH, like, for example, the enzymes of the stomach. They thrive in that very low acidic pH. Or others can do much better in a more basic environment, so a high pH. So this is enzyme dependent. So again, the Vmax is the maximal velocity of an enzyme catalyzed reaction. The Km is the amount of substrate required to reach 1/2 Vmax. This is kind of throwing a couple different definitions at you, but they're all referring to the same thing. We're going to go ahead and end this video here, and in the next one we'll talk about the enzyme inhibitors. Again, I want to make sure, just before, what I said about Km is 1/2 that Vmax. Don't worry too much about that. In the future, I may come back and rework that. Don't worry about that too much right now.